Hello, I'm Takuya Hirao. Today I will talk about transforming growth Factor-β1 and bone morphogenic protein-2 inhibit the differentiation into mature ependymal multiciliated cells. Ependymal multiciliated cells, here in F2 called ependymal MCCs, are lining on the ventricular surface and have essential role in cerebrospinal fluid flow like this. Therefore, dysfunction of ependymal MCCs, such as lack of cilia or ciliary movement disorder, causes CSF retention in resulting hydrocephalus. Recently, it is reported that Factor-β1 serum FBS inhibits the differentiation into ependymal MCCs in non-polarized conditions. However, regulatory factors involved in differentiation into ependymal MCCs have not been clarified. So the purpose of my study is to uncover the factors involved in differentiation into ependymal MCCs by using transfer permeable support filter, which is polarized conditions. Undifferentiated glial cells obtained from the whole brain of a newborn mouse were seated on the transwell. Transwell can distinguish two compartments. Upper chamber corresponds with the ventricular side, and the lower chamber corresponds with parenchyma side. In this study, undifferentiated glial cells were cultured in two different conditions. First is the FBS 3 minus plus which did not contain FBS in upper chamber. Second is the FBS containing plus plus, which contained FBS in both chambers. And I evaluated the effect of the FBS on the ependymal MCCs. Ependymal MCCs were visualized with anti-acetylcholine antibody, which is marker of cilia, and nuclei were visualized by DAPI staining. The percentage of MCCs was calculated by the ratio between the number of ciliated cells and the total cells. The ciliary movement was all observed using a microscope equipped with a high-speed camera, and the ciliary beat frequency (CBF) was measured as a parameter of ciliary movement. In the previous study, it is reported that the mean CBF was ranged to 11 and 20 Hz. So in this study, the mature ependymal MCCs were defined as over 15 Hz. Next is result part. This figure is the immunofluorescence images, and this one is the percentage of ciliated cells in minus plus and the plus plus conditions. In minus plus conditions, the percentage of acetylcholine positive ciliated cells increased with culture days. In the plus plus conditions, they also increased, but number of ciliated cells were lower than that of the minus plus conditions. Furthermore, in the minus plus conditions, the percentage of ciliated cells was about 60% at 28 days. But however, in the plus plus condition, the percentage of ciliated cells were significantly lower. Next, I observed the ciliary movement in the minus plus and the plus plus conditions. Beating cilia shown in red arrows and non-beating cilia shown in blue arrows were observed. In the minus plus conditions, almost all cilia were beating. The mean CBF was about 14 Hz, and over 50% of cells were differentiated into mature ependymal MCCs shown in pink zone. However, in the plus plus conditions, non-beating cilia were increased. The mean CBF was about 9 Hz, and only a few percentage of cells were differentiated into mature ependymal MCCs. From these results, it is clear that FBS inhibited the differentiation into mature ependymal MCCs. Among of several growth factors included in FBS, I focused on the transforming growth factor β1 as a candidate of inhibitory factors in FBS. 1 nanogram per milliliter of TGF-β1 was added in the upper chamber of the minus press conditions. This concentration is comparable with that included in 10% FBS. In TGF-β1 containing conditions, the percentage of ciliated cells were significantly lower than that of the minus press conditions. From these results, TGF-β1 inhibited the differentiation into ependymal MCCs like FBS. In the previous study, it is reported that 50 to 100 nanogram per milliliter of BMP2 inhibited the differentiation into ependymal MCCs in non-polarized conditions. However, the concentration in 10% FBS is about 60 picogram per milliliter. So in this study, from 60 picogram per milliliter and 100 nanogram per milliliter of BMP2 were added in the upper chamber of the minus press conditions. From these results, high concentration 1 nanogram per milliliter and 100 nanogram per milliliter of BMP2 inhibited the differentiation into ependymal MCCs. However, 60 picogram per milliliter of BMP2 did not inhibit the differentiation. Next, I observed the ciliary movement absence of the presence of the TGF-β1 and BMP2. The percentage of ciliated cells in these conditions were lower than that of the minus press conditions. Furthermore, the mean CBF were also significantly lower in both conditions, and mature ependymal MCCs were not observed. From these results, it is supposed that TGF-β1 and high concentration of BMP2 inhibited the differentiation into mature ependymal MCCs. Finally, to clarify whether inhibitory effect of FBS is recovered by TGF-β1 or BMP2 inhibitors, they were added in the upper chamber of the plus conditions. Each inhibitors alone, this one and this one, did not block the inhibitory effect of FBS, but two inhibitors were recovered. The ciliary movement also recovered by their inhibitors in combinations. From these results, it is suggested that not only TGF-β1 but BMP2 are major inhibitory factors included in FBS, major inhibitory factors involved in differentiation. In conclusion, FBS inhibited the differentiation from undifferentiated glia into the mature ependymal MCCs. TGF-β1 and high concentration of BMP2 also inhibited the differentiation Moreover, the inhibitory effect of FBS was not recovered by TGF-β1 or BMP2 inhibitors alone. However, it was recovered by their inhibitors in combination. This is the hypothesis of the inhibitory effect of FBS. TGF-β1 included in FBS inhibits the differentiation into mature ependymal MCCs like this. On the other hand, BMP2 may be secreted from the neighbor cells like ependymal cells or astrocyte, and secreted BMP2 activates the BMP receptor. As a result, it inhibits the differentiation into mature ependymal MCCs like this. From this hypothesis, it is indicated that the inhibitory effect of FBS can be recovered by TGF-β1 and BMP2 inhibitors in combination. Therefore, this study newly identified that TGF-beta1 and high concentration of BMP2 inhibited the differentiation into mature ependymal multiciliated cells from undifferentiated glial cells. That's all from my presentation. Thank you very much.